Okay, today we want to start our discussion of organic chemistry, and one of the things that we should mention before we start talking about some of the most important carbon-containing molecules is the fact that the reason carbon is such a versatile building block and the foundation around which all known life is built is because of its unique ability to form rings and chains, both with itself and with other molecules to which it can covalently bond. And this provides now a wide variety of molecules upon which to base life. Our first carbon-containing compound we're going to talk about are carbohydrates. They're made up of carbon, hydrogen, and oxygen in a very specific ratio, and that ratio is demonstrated here, where we have C and then in parentheses H2O, and then we follow this with an N, which indicates the number of carbons in that particular carbohydrate. So whenever you see a carbon-containing compound that has twice as many hydrogens as it does either oxygens or carbons, you can be reasonably well assured that this is a carbohydrate. The ratio of carbon and oxygen atoms is close to 1 to 1. The ratio of carbon to hydrogen is 1 to 2, and the ratio of oxygen to hydrogen is 1 to 2. As a result, you've probably surmised that this is a polar molecule, so it dissolves very easily in water, okay, due to its polar nature. And y'all know that sugars dissolve in water. That's not news to anybody here. We can divide them into monosaccharides, disaccharides, and polysaccharides. And it's the monosaccharides which are the simplest units of the carbohydrate family, and they can be assembled into polymers of 2, 3, or more sugars. A disaccharide would be a 2-sugar polymer, and a polysaccharide would be a polymer of more than 3 sugars. Okay, so we can see carbohydrates in any of these forms. They're important as a macronutrient, so they're an energy source, but they're also important in structure. They're found in connective tissue as part of the extracellular matrix that allows tissues to adhere to each other, and they're also obviously in secretions such as mucus, which provide a protective function for the body. And they're also important as ID tags. They serve as components of collections of molecules called antigens, which identify cells as belonging to a particular organism. And this is how the immune system distinguishes between an invader, a pathogen, and your cell's own tissue, so that it doesn't attack and destroy your own tissue and as a result produce disease. Lipids are composed mostly of carbon, hydrogen, and oxygen, and they're relatively insoluble in water because they're hydrophobic. They don't have that much oxygen in comparison to their carbon and hydrogen. So they're hydrophobic, they're non-polar. They function in protection, insulation, and physiological regulation. They're components of cell membranes in the context of phospholipids, and they're also an energy source. In fact, they're the most energetic of the energy sources in the macronutrients because they contain the most carbon-hydrogen bonds. And you have to remember that it's the carbon-hydrogen bond that packs the most energy of any bond for living systems. So the more carbon-hydrogen bonds you see in a molecule, the more biological energy it contains. Lipids can be either completely hydrophobic, in which case they tend to be transported in the body in the context of lipoproteins, or they can be amphiphatic, meaning that they have one part of the molecule that's nonpolar or charged, and another part of the molecule that is nonpolar and tends to associate more with other nonpolar components. So a charged or polar part and a nonpolar part, and it's in the context of the phospholipid that we see this type of molecule. Proteins are made up of carbon, hydrogen, oxygen, nitrogen, and occasionally iodine, and they do virtually anything you can think of as a life process. They regulate life processes, they aid in transport, they provide protection in the form of antibodies, also in the form of producing structural components that are part of our nonspecific barriers to infection. They're important in movements in the context of muscle contraction, and they provide physical structure for living systems, and they're also an energy molecule, although they don't have as much energy as lipids or as carbohydrates per gram. Nucleic acids are made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And examples of nucleic acids include ATP, which is the body's energy currency, as well as DNA and RNA, which are information molecules. DNA is found in the nucleus of every living cell, and it contains information important for everything that the cell does. Most of this information is dedicated to providing blueprints for the manufacture of proteins. These separate blueprints are called genes, which have their information transcribed to molecules of RNA when the cell needs that particular gene product. The RNA is transcribed from the DNA and then transported out of the cell nucleus, and here special cell organelles called ribosomes read the RNA and use those instructions to synthesize the protein. So these are information molecules, critically important for the function of the cell. Now, this is a table showing you some of the major functional groups of organic compounds. This is 2.5 in your textbook. Star this table. I want you to know it. I want you to know where these functional groups are found so that you can use them as a way of recognizing organic molecules when you see them. Let's talk a little bit more about carbohydrates. Simple sugars can either be 6-carbon sugars like glucose, fructose, or galactose, which are important in the diet as energy sources. And there are also 5-carbon sugars, which are vital components of the nucleotides, as well as DNA and RNA. The 5-carbon sugars are called pentose sugars. The 6-carbon sugars are called hexose sugars. Now, what you've noticed here in this diagram is that these sugars can take on a variety of different conformations. They can exist either as open chains or as rings. This is an example of an intramolecular reaction. It occurs within the molecule itself. Actually, I should say intramolecular reaction. And what happens when we go from the straight chain form to the ring form of the monosaccharide is that the hydroxyl group attacks this carbonyl group, and this is what forms, in the case of fructose, this 5-carbon ring, which has 1 oxygen with the rest of the positions being occupied by carbon. In the case of glucose, it forms a 6-membered ring, and in the case of galactose, it forms a 6-membered ring. So the sugars in an aqueous solution are in equilibrium between these open and closed chain forms. So you have to understand that that's constantly going on. These 3 sugars are examples of what we call structural isomers. This is a structural isomer between glucose and fructose. You can see that we have a difference here in this carbon position where the hydroxyl group is bound to the carbon. The difference here between glucose and galactose is that of a stereoisomer, where what we have is, again, the same molecules being bound to each other in the same order. The difference here being that the hydroxyl group in the straight chain conformation is going to project from one side of the molecule, whereas in glucose it will project from the other side of the molecule. Okay, so you have to understand the difference between these structural and stereoisomers. Shape is very important in living systems because this is how enzymes are able to determine what their substrates are. So it's not a minor thing when we have a difference in the way atoms are bound to each other. Okay, you can see here in the structural isomer that the structural formula is the same, but the order in which the atoms are bound to each other is different. In the stereoisomer, it's a matter of the orientation of the functional groups, which is different on a particular carbon. Okay. Disaccharides are simply the formation of a 2-sugar molecule from 2 monomers via a dehydration synthesis. Examples include sucrose, lactose, and maltose. These are common components in our diet. Sucrose is table sugar, lactose is milk sugar, maltose is found in a lot of dietary components that come primarily from plant material. Um, Sometimes, if we lack the enzymes to properly break down these disaccharides in the digestive system, they can produce complications. For instance, there are individuals who are lactose intolerant. Let's put a circle around lactose here. They lack the ability to cleave the bond that would exist between glucose and galactose. That's what the disaccharide lactose is made up of. As a result, this disaccharide doesn't get into the bloodstream of these individuals effectively because it can't be broken down into its monosaccharide components. The result is that it makes its way all the way to the large intestine, where bacteria, which normally live there, use it as a fuel source, and this produced gas and discomfort and terrible GI upset. Okay, problems with gastrointestinal motility, diarrhea, and so forth. Okay, so it's critical that we be able to break these chains of monosaccharides down into their monosaccharide component. That would be the reaction indicated by this arrow, using a water molecule to split them in order to use them as fuel in our diet. Okay, so we consume simple sugars, we also consume these disaccharides, and as you well know, we also consume the polysaccharides, which are long chains of many monosaccharides. These serve as storage molecules for monosaccharides, and they form part of cell surface molecules known as antigens. These are markers that indicate, again, to the immune system that a particular cell belongs to us. Glycogen is a polysaccharide that's formed by animals, commonly called animal starch, while starch and cellulose are formed by plant material. Starch in food is used as a source of monosaccharides for energy, while cellulose in food in human systems acts as bulk in the diet. We don't have the enzymes in our digestive system to properly cleave the glycosidic bonds that hook the glucose units and cellulose to each other. As a result, this polysaccharide proceeds through our digestive system unprocessed until it gets to the large intestine, where the bacteria can ferment some of this material, use it for fuel. It also serves to obviously add bulk to the diet, which allows us to more easily pass our stool. So this is why you've probably heard that you should have a lot of fiber in your diet in order to keep your bowel movements regular and to avoid such conditions as constipation. Okay. Now there are some animals that can actually digest cellulose and use it as energy. An example is the common termite, which has a bacteria that lives in its gut that secretes an enzyme that allows it to cleave cellulose into glucose, which now allows it to serve as an energy source for both the bacteria in the termite's gut and the termite itself. So there are some creatures out there that are capable of using this fuel source directly for energy. Humans, however, cannot. This is a table showing you some of the roles of carbohydrate in the body. Their structural role is important. They serve as components of extracellular matrix. They're also the backbone of RNA and DNA, as well as nucleotides. They're an energy source. The monosaccharides and the polysaccharides can be oxidized. That energy that's locked inside the carbohydrates can then be transferred to ATP, where we can immediately use it for cellular work. And of course, its role as fiber is important because it allows us to more easily pass our fecal material. If we didn't have adequate fiber in our diet, we would suffer from a condition known as constipation. Our next molecule is our most energy-rich molecule, and these are lipids. They're ingested and broken down by hydrolysis. One of the most abundant lipids in the body is the triglyceride. It's composed of glycerol, which is an alcohol that has three hydroxyl groups. You can see them here, here, and here, okay, as well as three free fatty acids, okay. The fatty acids are shown here, and it's the fatty acid component of the molecule that is the nonpolar part, so we'll indicate that in blue, okay. There is the nonpolar part of the molecule here. The part that I am circling. And then the polar part of the molecule, which will indicate in red. Okay, this is the polar part of the molecule. So you can see that most of this molecule's volume and most of this molecule's mass is occupied by nonpolar components. And so when we find triglycerides in the body, we generally find them as fat droplets contained inside a very special tissue known as adipose tissue. It's an energy-rich molecule, and the reason for that is that it contains lots of carbon-hydrogen bonds. In the diagram here, you've got 3 fatty acids that are relatively short, but most triglycerides are between 15 and 20 CH2 units long. And remember that it's the carbon-hydrogen bond that is the most energetic for living systems. As a result, you can see that fats are the all-time winner in terms of calorie, aka energy content. Fatty acids, which are a component of triglycerides, can be saturated or unsaturated. If they're saturated, this simply means that every carbon is bound to as much hydrogen as it possibly can in the context of the hydrocarbon chain. Whereas in unsaturated fatty acids, we occasionally will see double bonds, maybe more than one sometimes, in which case we call them polyunsaturates. This is an example of a polyunsaturated fatty acid known as linolenic acid. But again, you can see that there's quite a few carbon-hydrogen bonds in the fatty acid moiety, which is a part of triglycerides, and that's why it's the winner in the energy sweepstakes. Unsaturated fatty acids are better in terms of our overall health because they have less energy in them. As a result, when we cook with them, you're not loading up the food with nearly as much in terms of calories. In addition, the shape of these molecules doesn't allow them to easily adhere to the inside of blood vessels, where they can contribute to the condition known as arteriosclerosis. Trans fats are unsaturated fats that are artificially altered so that they have more carbon-hydrogen bonds. The problem with trans fats is that they take on unusual shapes inside blood vessels, and this allows them to contribute to conditions such as arteriosclerosis. Also, because they are saturated, they're very high in calories, and so if you eat a lot of trans fats, you run the risk of becoming obese, as well as the risk of accelerating the conditions such as arteriosclerosis. Now you might ask, well, gee, then why do we use trans fats in our food products at all? And the answer is very simple. These saturated fats have a longer shelf life, so they don't spoil nearly as quickly as their counterparts that have lots of double bonds. Unsaturated fats can easily go rancid, and the result is that the food you prepare with them can become dangerous to eat. Okay, so this is why trans fats are in a lot of the snack foods that you see in vending machines and in the grocery stores. Okay, now I told you that we were going to see the triglyceride in the context of the phospholipid, and here is the phospholipid. It has a hydrophilic head, which is made up of phosphorus and often a group that contains nitrogen. There's also quite a few oxygen atoms at this end. Remember that the glycerol molecule has 3 hydroxyl groups, which are bound to the carboxylic acid groups of the fatty acids in a dehydration synthesis. And so you've got a lot of electronegative molecules at this one end, electronegative atoms rather, at this one end of the molecule. As a result, this portion of the phospholipid is going to seek out water because that's the lowest energy arrangement of this molecule in an aqueous solution, is for this end of the molecule to seek out water, and then obviously for these nonpolar components to seek out each other. So other fatty acid tails will interdigitate, and that's part of what forms the phospholipid bilayer of cell membranes. That's the most low energy arrangement that those molecules can assume, provided we don't put any energy into the system. Okay, so this is their role. This is their structural role here as phospholipids. Now, eicosanoids are derived from fatty acids, and they're important signaling molecules. They include thromboxanes, leukotrienes, and prostaglandins. Incidentally, anytime you see a name, you should think about what the name means. Leuco meaning white, trienes. Leukotrienes are signaling molecules that act on white blood cells. The reason they're called trienes is because they have 3 double bonds in their fatty acid moieties, in the fatty acid tail that makes up the molecule. Prostaglandins are major mediators of inflammation, as are thromboxanes. There are also fat-soluble vitamins, A, D, E, and K, which we consume in our diet. These are nonpolar molecules that serve as tools for enzymes to perform certain metabolic functions. And this is why we have to eat our vitamins in our diet, so that metabolic reactions can occur. Okay, if you have an enzyme without a tool, you essentially have a non-functioning enzyme. So there's quite a few enzymes in the body that need these vitamins as cofactors in order for them to do their job. Because they are fat soluble, it's easy for you to overdose on them. So don't megadose on A, D, E, and K, because they can build up to toxic levels, especially in the fatty tissues in the body, and that can produce disease. The water-soluble vitamins, which are the B complex and the C vitamins, um, those are more difficult to megadose on, because generally what happens if you consume an excess of those, the urinary system will clear them. Okay, another class of lipid is known as steroids, and they have this very unique fused ring system where you have 3 fused 6-membered rings. They're carbon in all these positions where you don't have a symbol for the element. Okay, so this is all hydrocarbon here. And then 1 5-membered ring fused to the 3 6-membered rings. They are components of cholesterol, bile salts, and steroid hormones such as estrogen, progesterone, testosterone, estradiol, and so on. The carbon atoms in these rings are very hydrophobic. As a result, it's very easy for these molecules to cross cell membranes and affect the function of the cell on the cytoplasmic side. They're physiological regulators of body function, and they're also a structural component of cell membranes, especially in the context of cholesterol, which you can see here. Okay, this is cholesterol. Let's draw a circle around it. What happens when the cell wants to decrease the fluidity of its cell membrane, meaning the movement of the phospholipids along a plane in that cell membrane, is it'll put more cholesterol in it, kind of like a doorstop, to reduce molecular movement. And when it wants more membrane fluidity, it'll remove the cholesterol from the cell membranes. Okay, so these are important regulators of membrane fluidity. And you might ask, well, okay, why would the cell want to regulate its membrane fluidity? And if we visualize the cell for a moment, this would be the cell membrane, this would be the nucleus. One of the things that the cell membrane has on its surface are embedded proteins that do all kinds of wonderful things for the cell. They serve as transport molecules, they serve as cell recognition molecules, they serve as channels, they serve as pumps, all kinds of stuff. They serve as receptors, okay, and sometimes what the cell might want to do is move these molecules around so that they can aggregate, and as a result, what we would want to do is, once they're aggregated, decrease the membrane fluidity, and to get them aggregated, we would want to increase the membrane fluidity. So that's an example of how this might be used to regulate cell function. Okay, we'll get more into that when we actually talk about cells in the next chapter. So this is just a table showing you the role of lipids in the body. They're important in protections in the context of fat, which surrounds and pads the organ. They also serve to insulate the organs. The fat under the skin prevents heat loss. Myelin surrounds nerve cells and prevents electrical signals from leaking away from their targets. And they serve in regulation in the context of steroid hormones, which control a variety of physiological processes such as reproduction and secondary sex characteristics. They're also found in the context of the mineralocorticoids, which control electrolyte balance, and the glucocorticoids, which control sugar levels in the blood, just to name a few of the regulatory processes served by steroid hormones. In the context of vitamins, they serve as cofactors which allow a variety of metabolic processes to proceed. Vitamin A, for instance, forms retinol, which is necessary for seeing in the dark. It's a component of the the opsins in the eye, which are responsible for transferring light energy into electrical energy that the brain can understand as color and vision. Vitamin D promotes calcium uptake by the small intestine, which is vital for structural components of bone, but it's also critical for muscle contraction and nerve transmission. Vitamin E promotes wound healing. Vitamin K is important for blood clotting. They're also important structural components in the body that lipids serve their role. Phospholipids and cholesterol are important structural components in the membranes of cells, and of course they are a huge source of energy as a macronutrient. They can be stored and broken down later for energy. They have more energy per gram than either carbohydrates or proteins. Now, speaking of proteins, proteins are assemblages of amino acids, which are the building blocks of protein. When we build protein, what we do is we take amino acids and through a dehydration synthesis, we form a unique covalent bond called a peptide bond, which I'll circle here in red. This is the peptide bond. It only exists between the carbonyl of one amino acid and the amino group of the other amino acid. Incidentally, the reason that amino acids are called amino acids is because they have both an amino group, shown here, and an acid group, which is shown here. Okay, this is the carboxylic acid group. So that's where the name amino acid comes from. And you can see here in dehydration synthesis that we can assemble these amino acids in a long chain. That chain will then begin to adapt a shape, and that shape will be known as its native conformation once the chain is finished being built. And it's in that shape that the protein will serve its purpose in the living system. And the magic arrangement of these amino acids in the right order and in the right amount to produce these proteins that take on these shapes, that do all these tasks inside the living cell, is derived at as a result of the information that's contained in the DNA. Okay, so it's the DNA which has essentially the instructions for building these proteins. The DNA instructions are transcribed in the nucleus into RNA, okay, and the RNA then goes to the cytoplasm of the cell, where it is translated into where the information in the RNA is used to synthesize a novel protein molecule. Okay, so let's put protein out here. And this information in the DNA is the product of millions of years of evolution and natural selection. Essentially, the way this works, or the way that we believe that this has worked, is that these bits of information which are contained in the DNA molecule, if they serve the organism in terms of increasing its ability to live long enough to reproduce, what will happen is that that piece of information will be retained and will be passed on to the offspring of that organism, who will then pass it on to their offspring. And so as a result of combining these bits of information in a trial and error process that generates proteins that will either serve or not serve the living organism, we eventually arrive at a large collection of genes that defines that particular organism by virtue of the proteins that it manufactures. So it's not as if this information came about magically overnight. It took a lot of time and a lot of combining bits of information together until we produced something that increased the ability of that organism to survive that resulted in us holding on to that information and using it down the road. And which information served which organism best was really kind of up to the environment in which the organism found itself. Okay, so you can imagine that organisms, say, that were adapted for life in the water, or that evolved to be adapted for life in the water, would be the product of pieces of information that generated a body plan and generated a suite of proteins that were designed best for that organism's lifestyle, its ability to live and reproduce and feed in a water environment. Okay, same thing for an animal that lives on land or an animal that lives in the air. Okay, so it's a long process, evolution and natural selection. And if you're interested in a more detailed explanation about how this takes place, what you want to do is look up, Google Charles Darwin and Google theory of evolution, and it'll explain it to you in quite a bit of detail. We're looking here really at one of the finished products of evolution, or we could even say more accurately, an intermediate in an evolutionary process, the human body. So let's look a little bit deeper at protein structure. The general structure of an amino acid is shown here, with the amino group at one end, the carboxylic acid group at the other end, and this is the alpha carbon, to which is attached a hydrogen atom, and then a group called the R group, which will be any of 20 different collections of atoms depending on the identity of that particular amino acid. And you can divide these 20 different amino acids into three broad categories. You've got the nonpolar amino acids, whose R groups are primarily made up of carbon and hydrogen, so they tend to be nonpolar, they tend to be greasy, and they tend to associate with each other. You've got the polar amino acids, whose R groups contain electronegative atoms, things like sulfur and oxygen and nitrogen, for instance, and they tend to associate with each other or with water. And then you've got the charged amino acids, whose R groups contain nitrogen, often in the case of the positively charged amino acids, or carboxylic acid groups in the case of the negatively charged amino acids, C double bond OO. Let me draw a carboxylic acid group up here real quick for you. Okay, this would be a carboxylic acid group here. Okay, so those that have R chains that have carboxylic acid groups at neutral pH, they tend to have a negative charge. Okay, shown here. Let's get that bond complete. Okay, that's a carbon atom. I know it's not drawn real well. And those R groups that tend to have nitrogen-containing groups on them, amines, they tend to have a positive charge at neutral pH. And the reason for that, if you remember from our discussion on general chemistry, is because nitrogen has a lone pair of electrons in its outer shell, which can easily associate with a proton. And this allows us to form now an ammonium group, where this part is connected to the amino acid down here, and this part is connected to the three hydrogen atoms, and the nitrogen will have now a positive charge. Okay? So that's why often nitrogen-containing compounds tend to be bases, while acids, remember, are proton donors, so they tend to cough up protons. Okay, so there's the carboxylic acid group, and there is the amino group, and that's how we differentiate between the positively and the negatively charged amino acids. And so in the context of a protein, what do these residues do? They tend to seek each other out. Negative charges tend to look for positive charges, and vice versa. Okay. Often the charged amino acids are components of what we call the active site of the molecule. These are often found in enzymes. It's the active site where the chemical reaction that the enzyme catalyzes will often happen. Okay, so those are the general amino acid characteristics. as we assemble this long chain of amino acids together, the native conformation will be dictated to a large extent by these side chains seeking each other out. The nonpolar side chains looking for the other nonpolar residues, the polar side chains looking for the polar or the charged residues, and the charged side chains looking for side chains of the opposite charge. And that, to a great extent, dictates how the protein will fold up and assume its native conformation, which is the shape in which it's going to do its task inside the cell. Glycine is the simplest amino acid. The side chain for glycine is a simple hydrogen atom. So glycine is one of those funny amino acids that can be considered sometimes nonpolar, sometimes polar. Okay, it's again a very simple amino acid. Other examples of amino acids include tyrosine. Tyrosine has a phenyl ring. Okay, you might not have seen this shorthand before for the phenyl ring. It's basically shorthand for a six-membered flat carbon ring which contains conjugated double bonds. And you're going to have to forgive my art here because I'm drawing with a trackpad, but that's the six-membered ring. This is the same thing as that. Okay, the circle in the ring indicates that it's what we call in organic chemistry aromatic, meaning that these double bonds don't necessarily exist statically at these positions. They can alternate between the positions that I've shown and the positions that I'm going to draw here. Okay, and so the way we describe that in organic chemistry shorthand is we put a little circle here indicating that these are conjugated double bonds that can shift their positions from moment to moment. Okay, it's got a hydroxyl group on the phenyl ring. This is one of the building blocks of thyroid hormone, which is an important mediator of the body's basal metabolic rate. In the context of thyroid hormone, this phenyl ring is actually substituted with iodine atoms in place of the hydroxyl groups. There's two important thyroid hormones. One is called T3, which is short for triiodothyronine, and as you can guess, it has 3 iodine atoms in it. And T4, or thyroxine, which, as you can probably guess, has 4 iodine atoms in it. Okay, so those are that's one of the purposes for tyrosine. Tyrosine also serves often as an active site in enzymatic reactions. It's one of the active site residues that's involved in a lot of enzyme chemistries that do things like break down protein. Okay. Another role for tyrosine is obviously as a fuel molecule. Okay, it can be a component of protein that we eat in order to get energy. And there's a genetic condition called phenylketonuria in which certain individuals are born without the enzymes capable of breaking down tyrosine in the context of protein. The result is that tyrosine products build up in cells such as brain and other nerve cells, and this results now in the destruction of things like the brain and the spinal cord. The condition is known as phenylketonuria. Okay, and it's one of the things that we can check for using amniocentesis before the baby is born, and we can prepare the parents for a life with that disease. Individuals with PKU have to avoid phenylalanine in their diet in order to avoid the effects of the disease. One of the places that we find phenylalanine along with aspartic acid is in the context of NutraSweet, which is an artificial sweetener found in a lot of diet sodas. And as you can guess, first of all, aspartic acid is an example of a negatively charged amino acid, so I'll put a big NEG here. Okay, so at neutral pH this has a negative charge. In NutraSweet, it's the covalent bonding between phenylalanine and aspartic acid that produces aspartame and that's the artificial sweetener. It's kind of an unusual dipeptide, though, because it does not feature a a peptide bond in the traditional sense. Okay, so it's one of those things that we use to try and cut down on people's calorie intake, as opposed to drinking soft drinks that contain primarily sugars, carbohydrates that, as we already discussed, have a lot of calories per gram. Okay, so what do proteins do in the body? Well, it's probably easier to ask, what don't proteins do? Um, they're important in regulation. Enzymes control chemical reactions, and hormones regulate a lot of physiological processes. For example, insulin uh, affects glucose transport into cells, so it's an important regulator of energy uptake. Glucagon and insulin together regulate your blood sugar levels. They're also important in transport. Hemoglobin is a protein that transports oxygen and carbon dioxide in the blood, and there are plasma proteins that transport a lot of substances, things like hormones, in the blood. Proteins in the plasma membrane control the movement of materials in and out of the cell. They serve roles, for instance, as pumps or as channels or as ID markers. They're also important in protection. Antibodies protect against microorganisms and other foreign substances. You could think of an antibody as a magic bullet, kind of like a heat-seeking missile that always finds its target. The antibody will always find the antigen that it was built to bind to, and when it finds that antigen and binds to it, it marks whatever cell is displaying that antigen for destruction. Okay, so it'll go and attack and destroy that particular antigen and then wipe out that invader. They're also involved in movement. Actin and myosin are proteins that are important in muscle contraction. They're important structural components. Collagen and elastin are important because they form structural frameworks in many parts of the body. Keratin provides strength in skin and hair and in nails. And they're also important as an energy molecule. They can be broken down for energy, but per unit of weight, they're about on par with carbohydrates. They have maybe a little bit less energy than pure carbs do. Now, what we want to do next is I've got a series of videos here that will talk about how proteins are built, and then talk about the levels of structure in protein: primary, secondary, tertiary, and quaternary structure. So pay very close attention to this next block of videos. Take careful notes, because it's these levels of structure properly executed that define the role of the protein in the living system. Once the protein has adopted its native conformation, it's ready to go out in the cell and do its job. So let's take a listen.
Proteins are polymers of amino acid monomers. Every amino acid contains a central carbon atom bound to 1. An amino group, 2. A carboxylic acid group, 3. A hydrogen atom, and 4. A group that differs from one amino acid to another and is represented by the letter R. This dehydration synthesis reaction has created a protein consisting of 4 amino acid monomers.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function. In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself. Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups. Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
Okay, we talked about the fact that it's the native conformation of proteins that allow them to perform their proper task. This is just a little video that talks about what happens to a protein when it denatures, when it loses its normal shape, and how its properties change. So take a listen.
This familiar gelatin dessert actually is a good example of the process of coagulation of proteins into a three-dimensional latticework that entraps water molecules to produce a semi-solid gel. This animation explains the physical chemistry behind the process. Proteins are synthesized by polymerizing amino acids. The polymerization occurs by repeatedly forming peptide bonds that link individual amino acids together into a chain. There are 3 structural features that influence the three-dimensional shape of a water-soluble protein. Primary structure is the peptide bond between individual amino acids that creates a long chain of connected amino acids. These long chains of polymerized amino acids have hydrophobic, water-repelling, and hydrophilic, water-attracting projections that are oriented perpendicular to the chain, as shown in this illustration of a growing protein chain. Secondary structure is the helix that the protein chain curls into as a result of hydrogen bonds and other weak forces. Tertiary structure is created when the protein molecules fold back on themselves outside of the helical segments to put the hydrophobic portions to the interior and the hydrophilic portions to the exterior. Several helical regions can exist in different portions of the molecule. When the protein has folded and refolded to reach its most stable configuration, it will have mostly hydrophilic amino acid residues on the exterior and mostly hydrophobic residues directed into the interior. When natural proteins are subjected to physical or chemical treatment, their structure changes and they become unnative or unnatural. We call that process denaturation. In this example, heating the proteins in solution imparts energy to the protein molecules. This added energy is enough to break the relatively weak forces that hold the protein in its refolded and helical tertiary and secondary configurations. As the process of denaturation proceeds, the protein molecule unfolds more and more, and the internally directed hydrophobic regions now become exposed on the outside of the molecule. The peptide bonds are largely hydrophilic. Once these segments are set free from each other, they attract water molecules. The recruitment of water molecules entraps the water molecules in close proximity to the protein strands. The hydrophobic portions of the molecules are also exposed. This situation is unfavored because the hydrophobic portions of molecules are not stable in an aqueous environment. Hence, upon unfolding, the hydrophobic regions on individual protein molecules will associate with hydrophobic regions on other protein molecules. This situation encourages the association of these protein molecules into larger and larger random three-dimensional structures. The molecules aggregate into very large water-insoluble collections that are quite randomly assembled. As the proteins denature, latticework structures grow amorphously and attract the solvent water molecules into cell-like structures. The self-associated water molecules, in groups, adhere to the surface of hydrophilic regions of the protein, while hydrophobic regions of the protein dissolve into each other and provide the energy to retain the structure. As this process continues irreversibly, all of the protein molecules are recruited to this large insoluble mass in a randomly organized structural framework that contains entrapped water molecules. One example of the consequences of unfolding and reassociating protein molecules is coagulation of egg white. Frying an egg is no more complicated than denaturing the egg white protein. The assembly of irreversibly denatured protein molecules results in formation of a solid gel. The gel entraps water molecules inside the white into a semi-solid structure, which holds its shape under normal conditions. Other examples of denatured protein assembling into three-dimensional structures include the baking of yeast-risen bread, coagulation of meat proteins by cooking in such products as hot dogs, and in the solidification of gelatin upon cooling of a solution. Denaturation and coagulation of proteins is a complex, irreversible process. But the study of denaturation has allowed us to better understand the three-dimensional structure of native proteins.
Okay, enzymes are important because they lower the activation energy of the reaction that they catalyze. And what this allows the cell to do is to make the product quickly enough that it will serve that particular living system. Instead of relying on doing things like changing the temperature or changing the concentration of the reactants, what the enzyme will do is facilitate the collision of the reactants to make the product quickly enough for the living system to make use of it. The three-dimensional shape contains an active site where the reactants will attach, and often enzymes will engage in what we call induced fit, where by binding those substrates the enzyme will alter its shape in order to facilitate the reaction. Enzyme names usually end in ASE and often have the same word stem as a reactant. For example, a lipid is a reactant for a lipase, which would catalyze the hydrolysis of the fatty acid moieties from the glycerol backbone of a lipid. Okay? Cofactors are important because they're tools that the enzyme can use to perform its task. Now, not all enzymes need cofactors, but many of them do. They combine with the active site and they make the non-functional enzymes capable of doing their jobs. Organic cofactors are called coenzymes, whereas non-organic cofactors are simply called cofactors. Okay, so you can have enzymes, for instance, that need a metal ion in order to perform their task. You can also have enzymes that need one of the B complex vitamins in order to perform their task properly. This is why we have to have minerals and vitamins in our diet in order for metabolic reactions to occur. And what you're looking at here is just a quick video that talks a little bit about how coenzymes operate. So take a listen.
This animation demonstrates how B vitamin coenzymes must be present in order to enable some enzymes to carry out their functions. Many of the B vitamins assist in the complex biochemical processes that produce energy from food. The B vitamins are essential parts of coenzymes. Coenzymes are part of the machinery that allows reactions to take place. Enzymes are large protein molecules that catalyze biochemical reactions by lowering activation energies. Enzymes accomplish catalysis by configuring chemical reactants in the best possible spatial arrangement for reactivity. This enzyme catalyzes the release of carbon dioxide when producing energy from carbohydrates. Combining with the coenzyme helps the inactive enzyme achieve the appropriate three-dimensional configuration. For this reaction, thiamine must be present as the coenzyme. The green coenzyme molecule above is thiamine pyrophosphate, the cofactor needed to permit the enzyme and the substrate, pyruvate, to combine in the correct orientation to catalyze the reaction. Now all three molecules are nested together, and the substrate, pyruvate, combines chemically with the coenzyme, thiamine. The biochemical reaction can take place and energy, as ATP, and CO2 are produced as products of the reaction. Once this happens, the remaining complex, containing the two-carbon fragment, thiamine, and the apoenzyme, rearranges and releases the two-carbon fragment, acetaldehyde, and the ATP. The departure of the acetaldehyde opened the thiamine pyrophosphate to combine with another molecule of pyruvate, and the cycle continues. Meanwhile, the energy is now in the molecule ATP and is available to do work or to be stored. Another way that a coenzyme can influence the reaction between substrate and enzyme is to cause a change in configuration of the enzyme without interacting with the substrate. Here you see the conformational change in the enzyme needed to configure the enzyme to accept the substrate in a lock and key complex. This is called an allosteric control. The cofactor approaches the inactive enzyme and bonds to it in an enzyme-coenzyme complex. The new configuration is a good fit for the substrate. Once this enzyme cofactor substrate complex is formed, energy can be released as ATP, and the other reaction products can be released from the complex. Now the correctly configured active site on the enzyme is free to engage another substrate. The amount of B vitamins that are needed to serve as coenzymes is quite small. There is enough thiamine activity in 1.2 milligrams to meet the average person's needs all day. Extra B vitamins will not produce more energy. Excess B vitamins in the body will pass out of the kidneys into the urine. This table shows some details about several of the roles played by B vitamins as coenzymes.
Okay, the last important organic molecule we want to discuss are the information molecules, DNA and RNA, and their building blocks, the nucleotides. Nucleotides are composed of a 5-carbon sugar, a nitrogenous base, and a phosphate group. They include the nucleic acids, such as DNA and RNA, and energy molecules such as ATP. And you can see the building blocks here. DNA is made up of a pentose sugar, a nitrogenous base, okay, and a phosphate group. And these monomers are assembled in a long chain. There's usually two chains associated together in DNA in the form of what we call a double helix. The information that DNA contains is the result of the specific arrangement, the primary structure of the nitrogenous bases in the chain. Okay, so you can see here the different bases that make up DNA include thymine and cytosine and guanine and adenine. Okay, and as we'll see in a minute, DNA structure is very special because these two strands of nucleotides run in opposite directions. You can see that the building block, the nucleic acid building block, isn't symmetrical on all sides. You can see that there's a phosphate end and a hydroxyl end and a base end. And so we arrange these molecules in a 5 prime. This would be the 5 prime carbon here, okay, to 3 prime direction. This would be the 3 prime carbon here, okay. So this would be the 5 prime end. This would be the 3 prime end. And the other strand runs in the opposite direction. 5 prime end being up here, 3 prime end being down here. And the significance of this double-stranded structure of DNA is that the information contained in one strand is reflected as its complement in the other strand. And so let me show you what I mean by that. Essentially, for instance, if we have, say, a sequence on one strand where we have an adenine, another adenine, and then let's say, a thymine, and then maybe a cytosine here, okay. The other strand will contain what we call its complement, and the complementarity rules are that T always is going to be on the complementary strand to A, and G is always going to be on the complementary strand to C. So let's draw what that would look like here in the opposite strand. So we'll put green here. So the opposite strand would have a T here and a T here, okay, and there would be an A here, and then there would be a G up here, and so when DNA gets reproduced, when the cell gets ready to divide, it has to make a photocopy of its genetic information to have one copy go to each new cell. What happens is that these strands will come apart, they'll unwind, and the enzymes responsible for making the new DNA strand will use the information on one strand to recreate the other strand. And the result is that the information will be passed down, generally error-free, from living cell to living cell. Okay, so that's the significance of the double-stranded structure of DNA and what we call the base pairing rules, which are illustrated right here. That's why it's important as an information molecule. Now, when RNA is used as a method of getting the information in DNA out of the nucleus and into the cytoplasm, it does something very similar in that it will copy what we call the coding strand into a molecule of RNA, which will be a single-stranded polynucleotide. And this process, which is called transcription, creates this molecule called the messenger RNA. That messenger RNA molecule will then leave the cell nucleus, get into the cytoplasm, and then there are very important organelles called ribosomes, which will then read that messenger RNA from its 5 prime end to its 3 prime end and use those instructions to incorporate amino acids into a growing protein molecule. And when it gets to the end of the message, it'll let that protein go. That protein will then fold up into the shape it's going to take on in the normal cell, its native conformation, and then it'll be able to do its job. Okay, so that's the significance of these building blocks, why they're so important. The reason, again, that this part is called the nitrogenous base is because, remember, nitrogen-containing compounds are bases at neutral pH because they can pick up protons, and so they cause the pH of the solution that they're in to rise. Okay. In RNA, the pentose sugar is called ribose. You can see it's got hydroxyl groups here at the both the 3' and the 2' positions, and you've got here in deoxyribose at the 2' position a hydrogen instead of a hydroxyl group. Okay, so that's the main structural difference in the sugar phosphate backbone of these two polynucleotides. In addition, we should point out that in RNA, the nitrogenous bases are uracil, cytosine, guanine, and adenine, whereas in DNA it's thymine, cytosine, guanine, and adenine. In RNA, uracil is going to pair with adenine, and guanine is going to pair with cytosine. In DNA, thymine is going to pair with adenine, and cytosine is going to pair with guanine. So that's how the information gets passed. So DNA is the genetic material of cells. It's copied from one generation to the next. It's made up of two strands of nucleotides that run in opposite directions from each other, so they're said to be anti-parallel. Each nucleotide contains one of the organic bases of adenine or guanine, which are the purines, and thymine or cytosine, which are called the pyrimidines. RNA is similar to a single strand of DNA, but it's got 4 different nucleotides that make up the organic bases. It uses, again, uracil, cytosine, guanine, and adenine. And remember, again, that the pentose sugar in ribonucleic acid is ribose as opposed to deoxyribose. That is the pentose sugar. It's responsible for interpreting the code within DNA into the primary structure of proteins. And so what I want you to do is just take a listen to this video that talks about DNA and RNA, their relationship to each other, and their structural makeup. So take a listen now.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, The DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay, the last molecule we want to talk about is ATP, which is the universal energy currency in cells. It provides the energy for other chemical reactions, for processes that are anabolic, meaning that we build up large molecules from tiny ones, and it also drives movement. All energy required chemical reactions stop when there's inadequate ATP, and the energy in the ATP molecule, I'll circle the high energy bonds here in red, is here and here, okay? And you'll notice that the phosphate groups have negative charges. You can see them there and there and there. And what do we know about negative charges? Negative charges repel each other, and so in order to hold negatively charged groups together, it takes a lot of energy. When the energy is liberated from ATP, what happens is that we cleave these high energy bonds, and that energy is captured by other proteins and used immediately to do work. The work of movement, the work of synthesis, the work of growth and repair, you name it. The energy has to be in this form for the cell to make immediate use of it. While it's true that there's chemical potential energy in the food that we eat and the carbon-hydrogen bonds, even in the carbon hydroxyl bonds, there's energy in the carbs and in the fats and in the proteins. We can't directly take that energy and use it to do cellular work. We have to transfer it to ATP. Otherwise, we wouldn't be able to drive things like growth and repair and biosynthesis. And the process whereby that happens is called metabolism. And metabolism has, in living systems, has three broad pathways that are important to know. And I want to go ahead and introduce them now so that they won't be a surprise when we see them later on. They're glycolysis, which is just a fancy term for splitting sugar. And the sugar that is split in glycolysis is glucose. Glyco meaning sugar and lysis meaning to split. And then we have another very important pathway known as the citric acid cycle or the tricarboxylic acid cycle, the TCA, also known as the Krebs cycle. And then the last leg of metabolism is called the electron transport chain. So E minus for electron transport and then chain. The last two parts of metabolism take place in the mitochondria, which are cellular organelles whose job primarily is to make energy for the cell. The first part of metabolism, glycolysis, takes place in the cytoplasm. Okay, so this is cytoplasmic, and these two guys are in the mitochondria. The most bang for your buck actually comes from the electron transport chain, which makes approximately 32 of the 36 ATP that we can generate when we take a single glucose molecule and send it through all of these different pathways. Glycolysis gets us a measly net gain of 2 ATP per molecule of glucose, and we'll see exactly why that is when we get a little bit more into the chapter on metabolism. Okay? All right, again, make sure and review this material. Include it in your notes. We will be going over it, of course, live in lecture, and I will see everybody in class. Thank you.